Chapter 11, Section 2, Applying Mendel's Principles, Dihybrid Crosses. Previously, we've been talking about genetic crosses with one trait. Now we'll be looking at those with two traits, and it's called a dihybrid cross. The ones with one trait were called a monohybrid cross. So in the dihybrid cross, basically you cross, for the parental generation, you will cross two true breeding plants. And you're looking at two different traits on the plant. In this case, we're looking at round in yellow and wrinkled in green. So when Mendel crossed these, he had to figure out, we need to figure out, what's the genotype for the round and yellow peas. If they're true breeding, that means they are big R, big R, big Y, big Y. And then for the wrinkled in green peas, which is the recessive genotype, we're looking at little r, little r, little y, little y. Now, when you cross two of the true breeding peas that have two genotypes, they both come out heterozygous. As far as their phenotype, they're going to be round in yellow, and the genotype for the F1s are going to be big R, big Y, little y, which makes them heterozygous. So then Mendel crossed the F1 with each other to produce the F2. So we're looking at big R, big Y, little y, crossed with big R, big Y, little y. And these are the results that he obtained. So he got most of the ones were round in yellow. A few of them were wrinkled in green, and then quite a large amount were combinations of the two. So it would either be round in green or wrinkled in yellow. Okay? So what do these results tell us? They tell us that alleles for seed shape segregate independently of those for seed color. So you could have any combination of the seed shape. So you could have round or round in green, or you could have wrinkled in yellow or wrinkled in green. So there's four different phenotypes that you could have. Four different genotypes that you could have. And genes that segregate independently do not influence each other's inheritance. So if something is round, it can still be yellow or green. It doesn't have to necessarily be red and yellow. So his results were very close to this ratio, which is 9:3:1. And in this place, or because of this, Mendel discovered what's called the principle of independent assortment. The principle of independent assortment states that genes for different traits can segregate independently during gamete formation. So when you're looking at the alleles that are going to be in the gamete, it can be any of the combinations. So the inheritance of biological characteristics is determined by genes, and these are passed from the parents to offspring. That's one thing that we learned. The second thing that we learned is that when there are two or more alleles of a gene for a single trait, some will be dominant and some may be recessive. Okay. So in the 1900s, now remember Mendel was in when? The 1800s. An American geneticist named Thomas Hunt Morgan, he decided to use the fruit fly for his model organism. Do you remember what the characteristics were of an ideal organism? So before long, Morgan and other biologists had tested every one of Mendel's principles that he did with the pea plants, and Morgan and his people found out that this applies also to other organisms as well. So the basic principles of Mendelian genetics can be used to study the inheritance of human traits and even to calculate the probability of certain traits appearing in the next generation.